Hello, my name is Chris Fierro, and I'm a student enrolled in the Bio 515 course, or Physiology 1, at Grand Canyon University. Today's presentation is on the molecular basis of muscle contraction, or otherwise referred to as the sliding filament mechanism. Um, at the conclusion, I'll upload the video to this link right here on this front page. So here we see the transfer of a message. Okay, so going from the brain here, moving down and down the nerve to the skeletal muscle, and then eventually to the neuromuscular junction. So this is the site where a motor neuron and the motor end plate of a myofibril meet at the synaptic cleft. Okay, so right here. As an action potential travels down the axon, okay, so it's going to travel down the axon here. Calcium is released into the terminal end via voltage-gated calcium channels. So these calcium channels will be located around here-ish. And those, as the action potential travels down, those calcium channels open up. That calcium influx causes the excitosis of acetylcholine. So these vesicles right here contain acetylcholine, and that acetylcholine diffuses out to the synaptic cleft, which in turn opens some ligand-gated sodium channels. Okay, and once those sodium channels are open, those propagate an action potential that opens calcium channels on the SR, the sarcoplasmic reticulum, which is where the sliding filament mechanism begins. Okay, so here we have a muscle fiber, zoomed in quite a bit, and we can zoom in further on this myofibril right here to right here. Okay, and on this image, we can see a number of different things. So the sarcolemma, we can see the terminal cisternae, which is going to house that calcium. We can see, again, the sarcolemma over here. We can see mitochondria and the SR right here, the sarcoplasmic reticulum, and the T tubules. Okay, so we have a number of items here that are of note. Okay, so, and as we look at this, we can start to see these thick and thin filaments, which are going to be the actin and the myosin, okay, here on this myofibril. And as we continue to zoom in, here we can see the sarcomere. Okay, so the sarcomere is between the Z lines. As we can see by the image, the sarcomere is a space between the Z lines and the actin filaments. Okay, so it's these thin red ones. Okay, those are the actin. Okay, are gonna contract towards the M line, which is how contraction of muscle occurs. So, and again, here's an actin filament here. Actin filaments are polymers of G-actin molecules. Okay, so we have these G-actin molecules here. Okay, and they come together to form this F-actin strand. And on this actin filament, we also have two other proteins, the troponin, okay, seen here, and the tropomyosin, seen here. Okay, and as we zoom down to the myosin, and these myosin heads are able to articulate. They have this little hinge point, so they're able to articulate and move. So, and as we can see in this image, these myosin filaments are much thicker than these actin filaments. And another just point of interest is this titin, I believe is how it's pronounced, is it's another protein, but it has a lot of elasticity to it, which helps to give muscles elasticity. So here in this image, we have a broad overview of what is occurring, starting at the neuromuscular junction right here, once again. Okay, so again, once that action potential is propagated at the sarcolemma, it travels down the T tubule. Okay, so indicated here. Okay, it's this tubule here. Okay, and as that action potential travels, it causes the release of calcium um, through ryanodide receptors. Okay, RY, R1, I believe, for muscle, for skeletal muscle. So this calcium diffuses out through those channels or through that receptor. And it's this calcium that eventually comes and attaches to the troponin. And as that calcium attaches to the troponin, it exposes the active site because the tropomyosin is moved out of the way once that calcium attaches. Okay, and once that active site is exposed, the myosin head is able to form a cross bridge with the actin and be able to articulate it or move it. Okay, or slide it, which hence the name, the sliding filament mechanism. So on this image on the left, we see an energized myosin head, okay, as it has already hydrolyzed an ATP to ADP and a phosphate. We also see this actin filament, okay, with the active site blocked. Okay, the calcium cannot attach to any of these active sites, or the myosin head cannot attach to any of these active sites because they're not exposed. So calcium ions are available here, but they haven't attached or they're not bound to the troponin. Here, if we move to this image on the right, we see that the calcium has attached to one of the troponins and the tropomyosin has moved, okay, allowing for the exposure of these active sites here and here and here. Okay, so now this myosin head is going to be able to attach to that active site. So here in this image on the left, we see that this myosin head has attached to these active sites, or these myosin heads have attached to these active sites, and they're ready to slide. Okay, so now is when, and as I mentioned earlier, these myosin heads are already energized. So now that this cross bridge has been formed, the energized myosin head is able to slide the actin filament towards the M line. Okay, so this is moving towards the M line. It's going to slide it towards the M line. Here we see it as well. And that process is called the power stroke. And it's indicated by this arrow movement. So in this first image, we see that the myosin has used the energy provided by the initial ATP that provided the power stroke and is ready for another ATP. When that ATP attaches, okay, so once it attaches, the myosin head disconnects from the actin filament. Okay, so that cross bridge is no longer formed or no longer active. So, and here in this image, we see that the myosin has once again broken down that ATP via hydrolysis to ADP and a phosphate. So it is now energized and ready for that movement again. So that contraction can continue. So here in this image, we see the sarcolemma, okay, sarcolemma, and the embedded proteins that serve other purposes than what our current considerations are. So we have all of these other proteins that are embedded into the sarcolemma. Okay, but there's a couple of things that are of particular interest to us And the first one is this ryanodine receptor, which is the channel from where the calcium causing the sliding filament mechanism comes from as it releases stored calcium from the sarcoplasmic reticulum. Okay, so here we have the sarcoplasmic reticulum. This is where the calcium is stored. This channel is how the calcium leaves the sarcoplasmic reticulum. And then the other point of interest or thing to mention is the Circa pump, okay, or the calcium ATPase pump or the Circa pump. This pump is important because once muscle needs to relax, um, this is how the calcium is pumped back into the sarcoplasmic reticulum. So this, as mentioned, as noted in the name, it is very energy dependent. A lot of ATP is needed to be able to move this calcium back, so that's no longer going to bind to the troponin and continue this contraction process. Another interesting observation is from a DQ post that I posted today from a separate reading, is this pump is the most powerful calcium pump in the body or in eukaryotic cells, which I found super, super interesting. And here are the sources that I used for this presentation. Thank you very much and have a great night.